Okay, if everyone can please sit down so we can start the next session, that would be great. Okay, just waiting for my slides. Perfect. Okay, so welcome to the next session. The next session title is From Integrated COVID-19 Data to Integrated Pathogen Data Analysis. My name is Katharina Lauer. I work for ELIXIR. I'm the Senior Scientific Coordinator for Infectious Diseases, and I'm the scientific lead for the BiCOVID project. And I'll give you a little bit of an introduction to the topic, and then hand over to my colleagues who will talk about more different aspects of this topic. So this session is structured a little bit differently. We will leave the discussion to the end, and then you can either ask questions to each of us, or if there are no questions, I will grill the other three with a couple of questions. Okay, so, this is the cover of the Time magazine, May 2017. Warning, we are not ready for the next pandemic, and were they right? So just looking at a couple of the recent outbreaks over the last decade, infectious disease outbreaks are nothing new, and we'll probably will see more of them. In fact, if you go to the WHO website, look at disease outbreak news, you can see this is just a screenshot from September. We have yellow fever, we have Ebola virus outbreaks. So overall, we are always in an outbreak scenario. Just looking at the most recent COVID-19 pandemic numbers, so these are taken from the weekly report from the WHO between the 5th and the 14th of September, 3.1 million new reported cases, 11,000 fatalities. So as of the 11th of September, we have seen more than 600 million confirmed cases and over 6.4 million deaths globally. At the moment, the Omicron variant makes up most of the reported cases. So then what's very interesting, if you look through that report, you get a little footnote saying the following. Actually, I need to move a bit, otherwise I can't read it out to you. So the current trends describing the circulation of Omicron descendant lineages should be interpreted with due consideration of the limitations of the COVID-19 surveillance system. These include differences in sequencing capacity and sampling strategies between countries, changes in sampling strategies over time, reductions in tests conducted, and sequences shared by countries around the world, and delays in uploading sequence data. Okay, so this is interesting. The one resource data we base all our policies on is actually a limitation, and there is a big caveat. So let's look at those challenges in a little bit more detail. What are those data challenges? And I guess by now many of you have been involved in working exactly on those challenges over the last 2 years. So no universally adopted system or standard globally for collecting, generating, documenting, analyzing, and disseminating COVID-19 or any other pathogen data, really. We have fragmentation, non-compatibility of research data. There's an inability to integrate and link diverse data, and really preventing researchers from asking big scientific questions, like relating vaccine efficacy to specific lineages. It's really difficult, and it's really difficult to ask those questions across disciplines, let's say such as social sciences, public health, or molecular biology. We have seen regulatory bottlenecks, different interpretation of laws that were generated to actually make things easier, streamline things such as GDPR, different interpretation, probably every country you ask, every institute you ask, every person you ask. We have outdated IT systems and computational infrastructure, lack of expertise in sourcing, configuring, maintaining this technical infrastructure, and also no or very little standardization across analysis workflows and software heterogeneity. Right, we've seen it all before. This is nothing new. If you look back in the literature, here the Zika challenge, science, get over yourself, Zika data sharing should be the norm, challenges of data sharing in Europe in the European COVID-19 projects, here key issues from Sierra Leone during the Ebola virus disease outbreak. So really, why have we moved on so little? And what are these challenges and why are they so difficult to tackle? Okay, so what can we actually do as researchers and people who work on building this infrastructure to support European preparedness or support global preparedness through connecting and analyzing across country borders, connecting and analyzing data across diverse data types, and connecting and analyzing open and access-controlled data? And I'll talk a little bit about a project that is trying to address some of those aspects. The project is called By COVID, or Beyond COVID. It's a European Union Horizon Europe Research and Innovation Program funded project. We're now 1 year into the project. And just to give you a little overview of what we're doing here, the main objectives are to enable storage, sharing, access, analysis, processing of research data and other digital objects from outbreak research. So the focus here is COVID-19 as a use case, but pathogen agnostic in the future. We try to mobilize, expose viral and human data, link fair data, metadata, develop new tools for analysis and workflows, really to focus on the future pandemic preparedness, and overall try to embed everything in the wider European context. So just to give you an example of how we're doing this, really building infrastructure, bringing people together, and building trust. And this is all focusing on, well, we're trying at least to bring together disciplines, meaning we're working across social sciences, we're working with public health, and we're working with people who are based in the biomolecular field, coming together around a platform that will later be introduced by Coleman, the COVID-19 data portal, which was created by the European Bioinformatics Institute in response to the COVID-19 crisis. This is really an umbrella for an entry gateway for open research data, for access-controlled research data, various different types. This could be imaging, cohorts, this could be protein data, transcriptomics data. So you can see the data types are quite broad. What we're trying to do is really make this data accessible through this one entry portal and trying to connect and link the data in a way so we can ask those important questions that require us to work across disciplines. Okay, so overall, what we're trying to do is really strengthening of an ecosystem where researchers from diverse fields can plug in, have the opportunity to openly store, discover, mobilize, share, connect, analyze, and integrate data, really to enable repurposing and innovation. And doing this all with the COVID use case at the moment, but really having pandemic preparedness and pathogen agnostic approach in mind. And yeah, I've already said that. My 3 colleagues will give you now interesting examples of how this is going to play out on a more technical end. So we have Coleman from the European Bioinformatics Institute and also affiliated with the COVID-19 data portal that I mentioned previously. We have Bas, he's from the Erasmus Medical Center and very much involved in the VO project. And Wolfgang from the Universität Freiburg and the Galaxy project, who's looking more on probably the workflow and data analysis side. Okay, Coleman, if you want to make a start. Yeah, and as I said, if you can just hold all your questions until we've all presented. Okay, thank you very much and enjoy the talks.
All right, good morning. My name's Coleman. I work at the European Nucleotide Archive at the European Bioinformatics Institute, and I am going to give you a quick overview of the COVID-19 data portal, but also what that means in terms of the lessons we've learned about pathogen data sharing and what direction we plan on going to in the future. So I talk about pathogen data sharing at the European Nucleotide Archive, but really the ENA is just one small part of the COVID-19 data portal and one small part of what pathogen data sharing means for the future. So fair and open data for COVID-19 has really meant a lot for how we've managed to overcome the challenges of sharing data and what it's meant for overcoming the pandemic. Before COVID-19 hit the scene, there were already 32,000 sequences in the INSDC for other coronaviruses, which immediately gave a good jumping off point for understanding the relatedness of SARS-CoV-2 to other coronaviruses, but also a great jumping off point for its understanding its biology, which allowed us to go into vaccines, treatments, and then later, as we got better at sharing data, we were able to power analyses like variant reporting through open data. Lots of third parties were consuming open data to power the reporting, to power understanding of global evolution of SARS-CoV-2 over time. So the COVID-19 data platform is a platform for the open and rapid access to data and workflows specifically related to COVID-19. Built upon open standards and open infrastructure, the entire plan was to use what is already there and make it better and easier to access COVID-19 data. It covers the globe and it is accessible across the globe. There is no restriction on this data. There is no restriction on access. You don't even need a login to access the COVID-19 data portal. Everybody in every country sees the exact same view, no matter what, and that can be guaranteed. So that was launched in April 2020, and it has enabled a diverse range of research topics across COVID-19 because it's a biodata portal. It's not just nucleotide sequences, it's protein sequences, expression data, host data, controlled access human data. These are the things that matter, and you only have to go to one place to get it all. So it has several different components, but I'm only going to really be able to focus on about 4 of those briefly. And that would be the viral sequence, the access, the engagement and coordination, and the preparedness part, which we'll get to at the end. It's built across a number of foundations of different grant projects, but also a number of partners. So obviously, EMBL EBI being one of the core parts it's built on. We manage its deployment. But we've been working through the ELIXIR network of member states, the COMPARE grant, which preceded the current VO project, through which we have a number of technical collaborators, such as ELTE in Hungary, DTU in Denmark, and Erasmus Medical Centre, which Bas will talk about later on, but also a number of other grants like Horizon 2020 by COVID, and the EOSC Life Grant and ELIXIR Converge. So, when we're talking about the viral data, there's kind of 3 success stories that we've learned from open data sharing in COVID-19. Data mobilization is a big part of it. So throughout the past 2 years, we've had a country-by-country campaign across EMBL member states and ELIXIR member states, EOS, GB nations, so that we can try and help people get their data into the open space. And as a result, we now have 5.7 million raw read data sets and 6 million consensus sequences, which are kind of silly numbers if you really understand the scale of it. 5.7 million raw read data sets is over 25% of the raw read records in the ENA, of all raw read records in the ENA. 25%. That's 2 petabases worth of read information. This has been flowed into systematic processing at the European Nucleotide Archive in systematic variant calling and consensus sequence assembly. Those then feed into public health reporting to the VO project. These are monthly reports, again, fully open. Not only is the analysis open, but the reporting is fully open. These reports go to the European Commission every month. The viral data componentry, we kind of package as what we call the data hub system, where it's a data management system for users where they can share data privately and publicly, but also they can have access to analysis workflows that are completely open that they can then use if they do not possess the infrastructure to analyze their data themselves. And then on top of that, we have visualization tools as well. This is kind of a brief overview of the overall kind of workflow. So we set out with a goal early in the pandemic to analyze all public raw read data sets not really realizing it was going to get to 6 million odd data sets. So we're still catching up. But basically, there are three objects produced: unfiltered variant calls, filtered variant calls, and consensus sequences. Consensus sequences are kind of what most people would want. We archive those, we put them through Pangolin, give them lineages assignments, but they also power a custom phylogeny available through the portal. Filtered variant calls go into the European Variation Archive as well as Ensemble. The EVA accessions, the variants that are called, that way that if the reference genome ever changes, those accessions mean that you can still refer to the same variant. And then Ensemble can use that to power their variation browser and genome browser. Unfiltered variant calls is a big component. This is basically where you call everything, but the idea behind this was that people may want to, especially in retrospect, study for minority variants as they go backwards through time. See if there were circulating variants at a minority level in the population before they became dominant. And this powers our main visualization app called CoVio. All of this is archived back into the ENA and presented in the COVID-19 data portal in a completely open and accessible way. So the COVID-19 data portal is a massive platform now. It has over 20 million records across molecular platforms and literature. You can access all of the data resources and tools associated with those, as well as link out to the resources they come from. We have integrated search in the platform, so you can search one search term, search many records. It's obviously a web platform, but we have full API access and download tools associated with it. It's used across the globe for different things: the Galaxy project, Open Targets, the CRG Viral Beacon in Barcelona. Like I said, the VO monthly reporting. But when we're tracking our usage, we have 220,000 unique users and 5.6 million requests since we launched the data portal. A big part of this has really been our engagement with national coordinators. Brokering is a huge part, where we have one user is able to submit on behalf of many sequencing labs, and that's actually facilitated the huge part of the data mobilization platform. We have national data portals across 10 different states, but we also have working groups that are trying to focus on the metadata that's important to them going forward, like seroprevalence and wastewater. Now talking about kind of preparedness, we've tried to start extending work on the COVID-19 data platform to become a more encompassing pathogens platform. And while this work is ongoing, one of the things we were able to do over the summer was mount an agile response to the monkeypox virus outbreak, whereby we were able to present a focused view of monkeypox virus data within a week, but then we'll also have dedicated submission routes as well as supporting submission documents available for users. And there's currently ongoing work for processing all of the monkeypox data that's public and through analysis workflows so that we can offer the same systematic workflows and visualization as well. And really, the things I wanted to kind of say are what are the preparedness lessons, what are the future things we want to think about. And we're trying to build an all pathogens data platform, the same idea for COVID-19, but for every relevant pathogen. Not unique portals, but one portal for them all. So that when you want to find the data that's relevant to your pathogen of interest, you go to the pathogens platform and you find it. We want elastic open data systems that are able to respond to the needs of people that are trying to share data openly. And we want to have equitable data sharing so that the people who are accessing data and submitting data are doing it so in a balanced way, that nobody is particularly over-consuming or under-sharing. Depending on their ratios, which we have data to support that nobody actually does that. Everyone is a balanced submitter and consumer. But we also want to enable users to find data in an intuitive way. In order to be prepared, it needs for researchers to not have to dig to find the data that matters. It should be intuitive, and it should be easy to ask questions of the data so that you can do your work better in the future. Thank you.
We switched to, yeah, perfect. Thank you. Thank you very much, Coleman. So next in line is Bas. Bas is going to talk to us about using COVID-19 data genomics and sharing to set a new future standard.
Yes, thank you for this very kind introduction. I'm indeed Bas Oudemunnik. I work in the Public Health Virology Group at the Erasmus Medical Center in Rotterdam. And today I would like to tell you about what COVID-19 actually has done to our ability to share data and our policy to share data. And this will also set a new future standard, I think. So this real-time data sharing during the COVID-19 pandemic started already pretty early on in January 2020, where the first sequence of SARS-CoV-2 was shared by Chinese colleagues already on the 10th of January. And by now, I think over 13 million sequences have been generated and shared. And we have learned and shown that sequence data can answer a lot of different research questions. So it did not only allow for the rapid development of diagnostic primers and allowed for the start of the production of vaccines, we also started to establish sequencing protocols where we can use the genetic information of the virus to see where the virus did come from, how widespread the virus is already, how different is it from what we've seen before, is it part of an outbreak? So we did a lot of outbreak investigation in hospitals trying to see if there was actually patients to healthcare worker transmission and if it's part of a transmission cluster. And we could do this relatively fast because there was already a paper published in 2018 where they described a multiplex PCR method coupled to this nanopore sequencing where you relatively fast can generate whole genome sequences of certain pathogens. And we have now fine-tuned this protocol to be able to work with SARS-CoV-2. And because of this Arctic protocol or other protocols which are also circulating, we can relatively fast generate consensus sequences. And the nice thing about this is that this is a general method which you can use for different pathogens. So with regards to future preparedness, this method was originally described for Zika virus and other viruses, but we now adjusted it to SARS-CoV-2, and more recently even we got a protocol now to sequence the monkeypox virus using the same technology. And because of the machines which are currently using, which are the grid ion machines or the nanopore devices, they produce data in real time. So what we've also developed during the pandemic is a real-time data analysis workflow, where every time where new data is generated, it will be automatically synced to our compute system, and then it will perform some basic steps, like the demultiplexing, trimming, mapping, variant calling, variant annotation, variant filtering, consensus calling, and alignments, and lineage assignments. And this is also a nice system which we have developed in such a way that it is compatible for each of our pathogen. So we actually use the same method now to sequence monkeypox virus, but then instead of aligning it to a SARS-CoV-2 whole genome sequence, we align it to a monkeypox sequence, and the rest we can remain identical. And this method we actually used already in the beginning of the SARS-CoV-2 pandemic, so early in 2020. And then we had around 24 to 48 hours turnaround time between actually receiving the samples and generating whole genome sequencing and analyzing these results. And this was mainly due to the very fast way we could produce data and also the automation of the analysis workflow. So this is just a showcase where we used it for in the beginning of the outbreak in 2020. So we started the first SARS-CoV-2 detection was in the Netherlands, was confirmed on February 27th. And we found one additional case 1 day later. And we were able to generate 2 whole genome sequences of these 2 positive samples 2 days later. And then when we look here at the phylogeny, we could already see that based on all genome sequences of these viruses, we could see that these cases were most probably not related, but had a different origin. So they are likely unconnected and they are not from one source, which actually means that we already had 2 different introductions in the Netherlands within 1 day apart. And this is where we could use whole genome sequencing for. And later on, we did it more systematically. So we did the systematic sequencing during the exponential growth phase. And there we actually sequenced day and night. So we also worked in the weekends, and we just kept on going to try to produce as much data as possible, which you can also see now here in this phylogenetic tree, because this actually contains all the sequences generated, or it's a zoom in of all the sequences generated from around the world. And at a certain point, around 20% of all sequences were derived from the Netherlands. But we could see that what we could mainly use this sequencing data for is that the increase in COVID-19 patients, as well as the increased affected geographical areas and the occurrence of local clusters, provided support for the increased movement restrictions. So we could actually use genomic information to try to predict how widespread this virus already was in the Netherlands. And coupled to the right metadata, we could see that the virus was already more widely spread than anticipated. Also because at that time we could not perform diagnostics on all people who had complaints, only on healthcare workers and patients admitted to the hospital. So that's why we then used whole genome sequencing to have a better idea about how widespread the virus already is in certain countries. And something about the data sharing policy. So there are two main repositories where we at least submit our data to. So we submit our consensus sequences to GIS8, which are at the moment over 30 million sequences generated and shared with the public. But this is not really completely open data, so you need to sign up and you need to have a username and a password to be able to access the data. And also you're not allowed to do anything with the data you want, because you have to sign a user policy, which you have to fulfill, because otherwise your access to the GIS8 system will be discontinued. And then there's the ENA system, where Coleman also already talked about, where there are now around 5.6 or even more data sets submitted. And just to illustrate the difference, because GIS8 only works with consensus sequences at the moment. A consensus sequence is a very small file, and we submit all our raw read data to the ENA, and there you talk about very large data files and also just raw data sets. So these still has to be analyzed, and here they can just directly work with your consensus sequence. I have to say this one works a little bit faster, because generally it takes 1 hour before your data is indexed and processed and everything is available to the users. But you can also imagine that it takes more time if you submit very large data sets. I mean, even the upload can take longer than a few hours if you submit large batches of sequence data. But this is then completely open data. And this data is also now used in public websites to monitor the genomics of SARS-CoV-2. So we have, for instance, we have the GIS8 systems where you can search and look for specific mutations and specific things you want to look for in the database. So you can look for different variants, you can look for specific spike mutations, you can look for a whole range of different things. And then this is also, at least it was coupled to the next strain data sharing system, where you can actually see the phylogenetic data of all of the SARS-CoV-2 sequences submitted to GIS8. And then on the other hand, you have the COVID-19 data portal, where a lot of raw read data are being submitted to, and which you can also search and find interesting signals there. And then there are two main websites who build upon the data present in either the COVID-19 data portal or in GIS8, which is, for instance, Outbreak Info, where you can nicely visualize the occurrence of certain lineages during time. And then there's the COV Spectrum websites, where you can also monitor the sequencing density, but also the occurrence of certain mutations, and they also try to predict what it might mean in the future. But these are all built upon the two repositories which I mentioned previously. And why is it important to keep on monitoring and tracing SARS-CoV-2? That's because all viruses change over time. We know that they are mutating. So we already expected in the beginning that we would see new variants emerging. But we also realized that most mutations have no impact on the function or the structure of the virus. But some can even be harmful for virus survival. But sometimes you have some mutations which can be beneficial for the virus. And this can mean increased transmissibility, increased disease severity, or an influence on the efficacy on vaccines or therapeutics. And we've also seen that the chances of these mutations will increase with the frequency of human and animal infections. And mainly in animals, we saw that the virus can be spreading massively, for instance, in mink farms or in wild-tailed deer in the United States. But despite that we have this massive genomic surveillance now in place for COVID-19, these variants of concern, of these variants of interest, still came a little bit out of the blue. So even though at peak points we sequenced close to 500,000 sequences of SARS-CoV-2 every month, still we see the occurrence of these variants of concern, which seem to come a little bit out of the blue, because they are almost 25 to 30 nucleotides different from everything which we've seen before. So this really raises now the questions to us, like where these SARS-CoV-2 variants of concern originate from. And there are three main theories on this emergence of these variants of SARS-CoV-2. And the first one is that the virus might have evolved gradually in parts of the world where there's less genomic information, but there's still widespread circulation and only limited travel, because it takes some time before all of these mutations can accumulate in a certain population, and you do not find it back in places where you do have active genomic surveillance. It might be an unknown animal reservoir, And that's also based on the findings now in the Americas, that these wild-tailed deer have been infected for around 2 years already, and they keep on spreading the virus across each other. And actually, you can find all different SARS-CoV-2 variants of concerns and different lineages in wild-tailed deer. Or it can be that there's a persistent infection of SARS-CoV-2 in an immunocompromised individual. And I'll explain this a little bit in more detail. So we recently received a sample in our clinical surveillance system, where we certainly found a variant which was not circulating in the normal population anymore. So all of a sudden, in the Omicron era, we've still found a patient infected with the Alpha virus variant, which was quite remarkable. But later on, when we contacted the submitting party, we asked them if there was something special about this certain patient, because we found an Alpha virus variant which we didn't expect to find. And then we could see that this virus was actually coming from an individual with dementia and with a chronic lymphocytic leukemia, so was severely immunocompromised, who turned out to be infected with SARS-CoV-2 for close to 300 days. And we actually were able to retrieve some samples from previous time points, from the 13th of April to 26th of May 2021, and then this particular case of the 31st of January 2022. And we could see that all of these viruses belonged to the Alpha variant, which had not been detected anymore in the Netherlands since somewhere halfway October. And then when we looked at the mutations which we could find in this immunocompromised individual, so all the way here below is the earliest time point, and then above are the 3 later time points. We can see that this patient accumulated several mutations in the genome of SARS-CoV-2, which was infected in the patient. Phylogeny also showed that this was most probably a recurrent infection, so it was just an ongoing infection rather than a new introduction or a new infection. And we could see that especially in the spike protein, there were quite some mutations present. And of these mutations, several are known to have an impact on the immune evasion of SARS-CoV-2. And for instance, here 3 mutations are located in the RBD region, close to each other, which is also an important part of the receptor binding of SARS-CoV-2. And we can see that 2 of these mutations are, for instance, also present in other variants of concern, and in this case, in Omicron BA1 and BA2. And then to start off with, also a little bit about monkeypox, because we've seen with SARS-CoV-2 that sequence data sharing and analysis was very important for public health decision making, and it was quite essential to see how the virus was spreading throughout the population. So we've learned from that, and we also started to early share monkeypox viral sequences. So also here we tried to produce data in real time and share the data in real time. And here, for instance, there's a virological.org website where people just post a small story around their consensus sequences, so where actually the sequence was coming from, after they submitted the sequences already to public databases. But then they described the first descriptive study, how they generated the sequence and where it came from. This is not peer-reviewed, so it's just a blog, and people can just submit actually everything what they want. But the most striking thing is then that also people can do subsequent analysis. So you have dedicated groups who produce the sequence data, but also groups who then are expertise on, or have more expertise on phylogeny and more detailed analysis. And we could see that they already pulled together all genomes which were generated and shared on this platform, and did some initial observations and did some initial studies. And actually here they saw that most probably monkeypox was already circulating among humans for a longer time than anticipated, based on certain mutations in the viral genome. And then there's also, they directly generated a GIS-8 repository for EPpox. As Coleman explained, also the ENA published a dedicated monkeypox website where people could submit their monkeypox sequences to. And this is actually quite new, because in the past we did not used to do this. We just used to first generate our data, write a report, publish it in a peer-reviewed journal, and then submit the sequences to public databases. I think that's now really shifted, as you can also see now for monkeypox since the May 2022, almost 1,900 sequences have been generated and shared in the open domain. But this is really not a given, because still before the pandemic, we did not used to share sequence data because you want to protect your intellectual property, you want to do your own analysis, you want to write your own manuscripts, and then later on share the data with other collaborators. But we've seen now that this really changed during the SARS-CoV-2 pandemic, because it's really appreciated that you share your data in real time. It just still questions who has the right actually to publish the data you generated. Is that the persons who analyze the data, the persons who generate the data? Preferably that should be merged and they should publish together, but that's not something that happens. So speaking from personal experience, we got scooped 4 times based on our own data set because we actually shared it with the population, and then other people started to analyze it, write manuscripts, publish it, and then, yeah, our paper got rejected since it was already published. And we were actually busy trying to generate more data and try to make more sense out of it, while other groups just focus on the analysis of that data. Can, of course, progress faster. But it would have been nice if they would have included this, or at least let us know what they did with our data. But it is a nice thing with the improvement of sequence technologies that it's really possible to monitor the emergence and evolution of the virus close to real time. There are some lags there, because in the beginning we did it indeed within 24 and 48 hours. But that meant that we have to work in the weekend during the evenings, and it's quite a burden on the lab personnel. So now we are a little bit slower, and we try to upload at least once a week to the public repositories, but we don't do it at a daily basis anymore. And something which we also realized during the pandemic is that it's quite important to not only protect the intellectual property of the data generated, but also that it can have quite some impact if you publish your data and share your results publicly. And one example where South Africa is still quite angry about is that they have the best surveillance system in the entire African continent. So they were actually the first one to detect a novel variant. And how we reacted here in the West is that we said we are not going to fly anymore to South Africa. There's no tourists anymore. So this actually had quite severe consequences for South Africa. But in the end, it turned out that the origin of this novel variant was most probably not South Africa, but somewhere else in Africa. And then we actually punished the ones who are sharing the data and who are open about it. And I think that should not be the solution. And I would like to thank all of these people, and thank you for your attention.
Okay, thank you very much, Bas. And we move on to the last speaker, which is Wolfgang. And he will talk about fully transparent viral genomics data analysis and sharing for everyone using public infrastructure.
So, yeah, thanks for having me here, Katharina. Yes, to put my talk a little bit in perspective with regard to previous speakers' talks, the ELIXIR project that I'm representing here is really a worldwide effort that tries to bring computational capacity and scientific analysis tools to as many users as possible through a graphical user interface and a consistent framework. Most notably, different teams in the project around the world are operating the huge public ELIXIR servers that many of you might know or might have used already, like usegelixir.org or the European ELIXIR server. But the project is also about, maintaining and developing the open source code of ELIXIR that allows you to set up ELIXIR in-house in your department, institute, or organization, and about developing tools and scientific workflows that allows our users to do meaningful analysis on these servers. And so with the SARS-CoV-2 pandemic, me and many people in the community have started to produce high-quality, reproducible workflows with the aim to bring these workflows to as many end users as possible, be it virologists, epidemiologists, or bioinformaticians, and to empower as many users as possible to analyze their data with a standard reproducible workflow. So me personally, I'm working in the ELIXIR Europe team, so the people behind the European ELIXIR server, and my focus is on this aspect of tool development and scientific workflow development. So where does ELIXIR fit into the picture of SARS-CoV-2 genome surveillance, for example? We've worked on many different aspects of SARS-CoV-2 genomics, but this has turned out to be currently the most dominant one with the most public interest. So I'm sure pretty much everyone has seen these plots like this on the right here by now, so showing the frequency of observed lineages in different national genome surveillance efforts over time. And yeah, you can see that all too familiar situation right now where wave after wave, previously dominant lineages get replaced by new ones. In light blue, you have BA5, which is right now the dominant lineage all over the world, with the notable exception of India, which I don't have on the slide though. But what's less known to many people is maybe how you arrive at these plots. So how do you get from a SARS-CoV-2 patient sample to and the sequence reads from it to these plots, right? And so previous speakers already showed you that what's mostly used nowadays is this tiled amplicon approach, where you take nasal swab from a patient, you amplify the genomic material in it with a set of 100 primer pairs approximately. And then when you sequenced these mixed amplicons, these combined amplicons, you get read information about the full viral genome. But then obviously you need lots of data processing steps to transition from this initial set of viral reads to these genome surveillance information at the high level. And one key feature of all these data processing steps that I'd like to point out here is that they are associated with data reduction and information content reduction, and a dramatic one. So essentially, they take as these processing steps take as input these sequencing reads, which might be dozens to hundreds of megabytes of data, and they reduce it first to a viral consensus genome of the isolate, which is around 30 kilobytes in size, and then further to just a short string that represents the lineage information, right? And so there's a high risk here if you do not do this data processing in a standardized way of fragmentation, so every group running their own combination of tools here with custom parameters and whatever, and so you can have thousands of combinations of tools and different analysis workflows. And that in turn would of course lead to poor comparability of results, and so in the end, across different such projects, even in different regions or different departments, those observed frequency might not mean as much as they could. Also, if you don't keep the original data, then of course you prevent any follow-up. That's a point that Coleman made already. In-depth analysis, retrospective analysis of the data, looking, for example, for minor variants in the data. And to prevent all this, we have worked on workflows that would allow you to do reproducible data analysis from these sequenced reads up to isolate consensus sequences, reports, and lineage assignments. We do release defined releases of these workflows through public registries. All the development of these workflows is happening in a version-controlled public manner. All our workflows produce high-quality intermediate data in standard formats, so like, of course, BAM format for mapped reads, VCF for variant calls, and they are ready to be executed on public infrastructure, but also in-house infrastructure on your own Galaxy server if your institute has one. They are easy to customize through a graphical workflow editor, so it's easy for you to adapt our workflows to your in-house specific needs. And they are highly scalable, from like dozens of samples per week in a small department up to really the needs of national genome surveillance projects. That's the workflows we've built, so the things in the red boxes are workflows. I won't go into detail here. You can ask later during the discussion round. but it's pretty much what Bas already showed for his SnakeMake workflows in their department. So mapping, producing VCFs, then consensus variants, and some batch level quality controls. So if a whole sequencing batch maybe suffers from a sequencing library preparation artifact or the like. What's more important is that all these workflows are available, and all releases of those workflows are available on the two major public workflow registries right now, on Dockstore and on Workflow Hub. So you can go there and pull any version of any of these workflows you like. You can also, from these registry, directly bring these workflows into your Galaxy instance of choice. So for example, from the Workflow Hub, you can choose to run any release of our workflows directly on usegalaxy.eu. If you do that, you're getting redirected to Galaxy Europe with the workflow run interface already set up for you, so you can run it directly on your collection of input data. Together with this, the workflow will also end up in your list of workflows you own on that public Galaxy instance, so that you can reuse it to run it on later batches of additional data. And you're even getting notified with this little green check mark here that this is a defined version that you pulled from a public workflow registry, and this check mark will disappear when you modify the workflow. So you can say, oh, that's not exactly what it should be, that this is something customized by somebody. Yeah, and as I said, this is all ready to be executed on public infrastructure, and with public infrastructure, of course, I mostly mean the big three public Galaxy servers in the world, usegalaxy.org, the European Galaxy server, Australia, but there's many more, like the Belgian Galaxy server, which is also open to the public. Yeah, and all these are powered by quite expensive and good public infrastructure. These are their addresses. And by the way, before anyone starts Googling about all these efforts I'm talking about, I have a last slide that collects all necessary links for you, so you can just take a single picture of this last slide. And yeah, it really allows analysis at any scale, and very conveniently, Galaxy will handle all the scheduling, parallel execution that's necessary when you have a batch of, say, 100 samples to analyze for you transparently in the background. No need to care about IT, admin, things that are really not the job of a biologist or a virologist, but you can focus on your analysis. Now, if you do that and you do all your analysis within Galaxy, then of course it is convenient to be able to submit your data directly out of Galaxy to the European Nucleotide Archive, and that's something we really want to push. I mean, it's important that this data doesn't stay in-house, but that it really gets to a public repository, and the ENA is a very good partner for that. So we have a Galaxy tool that allows you to do just that. You can fill in all the metadata about your sample and then upload your sequencing data directly out of Galaxy to the ENA. So all these additional steps of downloading it to get it out of Galaxy, then submitting it with a command line tool or web interface to the ENA again, that's not necessary anymore. And then, yeah, I mean, we could have stopped there, but then we thought, okay, instead of just saying, hey, there are these workflows, you can use them and they should work for your samples, we are actually continuously testing this through a large-scale project we set up. So we did a lot of automation through scripting, basically using the Galaxy API, where we now pull regularly data from the ENA, from the public sequencing data archive of the ENA, raw read data, from different projects that we are very confident about the metadata and sample quality. We're running this through the published releases of our workflows to produce all key files of an analysis, BAM files for the mapped reads, VCFs for the variant calls, and faster consensus sequences. And we're depositing them on an FTP server that is kindly provided us by the Barcelona Supercomputing Center. So you see the name also down here. So I'm finally close to where the data ends up that we're exporting from Galaxy. That's quite nice. And from there, we're actually acting kind of as a data provider for this data for different downstream consumers. So the Viral Beacon project, the UCSC genome browser, so there's a Galaxy track for that now. And we also have our own observable dashboard where we can visualize our own results. And we have currently high-quality analysis results for around 400,000 samples. That's just a fraction of total data available at the ENA, of course, but from around five different countries. But as I said, this is data that we're really familiar with, the metadata that we can analyze in the best possible way, where we know how the batches are grouped and so on. Good. And with that, just what's next? I mean, that has been discussed extensively. We need to be prepared for the next disease, and this means maintaining the existing workflows, the tools behind them. That's a lot of effort, actually. Adapt workflows to other pathogens of concern. We have done this already for our Galaxy workflows for monkeypox. So on Workflow Hub, you can also pull a monkeypox workflow. What's currently work in progress is benchmarking really the effects when you do a workflow change. How does that impact analysis results in the future? This is what I'm currently focusing on in my own work. And of course, you need to maintain infrastructure, framework services, build Galaxy servers. And if you are not invested yet in these things as a virological department, then you should consider this, right? I mean, the next outbreak will not produce less sequencing data, but probably more. Good, and this is the slide I had promised with all the links, so I just leave it up for you to take notes if you're interested. Okay, and...
Right. Trying not to fall over. Right, so we started a little bit late, so I'm taking a little bit of your break, I'm afraid, to have a little bit more of a discussion, so if you could all come up. Right, so I think I'll just start off by asking the same question to all of you, and I think you need to group around the microphones, otherwise people won't hear your answer. But I guess one of the burning questions that everyone in the room has, as a bioinformatician, what do I actually need to do now? And I guess, Bas, you will probably say, please recognize our work, give us credit for our work, and then we're more likely to share. Or what would you say to someone who says, what can I do to actually move things forward?
I think actually GISA has a really nice user system in place, and also certain constraints and things you have to do if you want to use the data of a group who published this data. So it can be, we try to explore several different ways. So one thing which we might use is a data DOI. There you just get a citable set of data which you can just cite in your journal, so you will be recognized for the work you've did. And yeah, it's also not easy. If they just take only your sequences and just publish only the sequence you have generated, is different than if you combine all the data generated by different partners. Because of course, I mainly sequence viruses from the Netherlands. If you want to do a Europe-wide study, I can imagine it's very difficult to include, how many countries do we have? Mm-hmm. Quite a lot of countries. And they all want to have 2 or 3 different authors on the manuscript, then it gets complex.
So I bring Coleman in now, and I know that the EBI uses, for example, links data to ORCID IDs. How do you do that? Is that something that will help in the future to get people to share more?
Yeah, so recently we did implement a system where you can now claim projects and data sets using your ORCID ID, which gives people a much clearer way of attributing how they contributed to public data sets, particularly, because that gets added to your ORCID record. You can add that more easily to your CV. And then that also becomes a kind of linked way for you to understand if someone cites a project, you can see that someone's cited a project that you're involved in. It gives people a bit more of a transparent way of getting credit for projects they've contributed to.
Okay, Wolfgang, I'll ask you the same as well, but a little bit a different angle. Looking at the data analysis side, workflows, how can we give people credit for putting those things together?
Yeah, I mean, one thing is, of course, through the workflow registries that I already mentioned, and if you have a defined release, of course, you can be a creator and author, can be acknowledged as a creator, author of these workflows, and you can also assign document identifiers to workflows, and just like scientific software, you can also cite workflows this way, right? So it's an important development.
Okay, fantastic. Frederick, you want to ask a question?
I have a question on the data mobilization. We did a lot of effort, as you mentioned, and we have a lot of data, but it's not a very uniformly spread data set. I mean, there's the UK that has a huge bias, let's say. Within Europe, there's biases. Globally, they're even worse. What can we do to kind of improve this situation long term?
Everyone was nodding, so who wants to go?
I think from my perspective, one of the biggest things that we kind of want to address in the future is building trust with global South nations, because they don't really feel like equitable partners in data sharing. Often they run the very real risk of global North countries engaging in parachute research when they share data. They just take that data, run with it, and publish interesting results. There's no immediately good solution for that, and what I would like to see is an investment in helping build infrastructure and build bioinformatics centers in global South countries so that they can become those strong, equitable partners in data sharing, and they feel like they are part of that global community and can feel like they can contribute as much.
Thank you. Do you want to add anything, or has Coleman covered it?
I think it's also quite complex to actually generate the data at these sites. For instance, I've been to Suriname. I tried to do capacity building there, tried to implement SARS-CoV-2 genomic surveillance in Suriname, but they are really not up for it. They first have to focus on diagnostic tests. They have a Windows computer from 2000, and they simply don't have infrastructure to do it. So I think it's also a larger problem that we should invest in infrastructure and these kind of things in certain countries.
I'll keep you here because we had someone actually type something in through the pad. He asks if these surprise variants happen, right? How can we quickly translate that into policies and clinical practice? So what's the approach here?
A really good question, and I think that's something I will be working on in the next 10 years.
Okay.
How we can also
so you'll have a job?
We just have a sequence. We don't know what it means. So how can we translate this genomic sequence, perhaps with artificial intelligence, these kind of things, to have some sort of a phenotypical impact, that we really know what these variants are doing, rather than waiting for it to happen.
Okay, I see we have another question from the audience.
It's building on the previous question, the trust for sharing data. Fantastic, but I mean, we have been hitting legal grounds. We have been hitting problems in Spain within regions, across borders. I think it's much more than trust, and that we should be working in suggesting legal ways to do something.
Wolfgang, I guess you guys from Galaxy have experience with that.
I mean, this is certainly true when it comes to patient data, like when a sample got collected exactly and the location of it and so on, and maybe gender and these kind of things. But there's a lot you can still improve typically on the metadata within legal bounds. Yeah, so something that's essential for good bioinformatics analysis, for example, is really those technical details, which samples come from which batch of sequencing have been processed together during the preparation of the sequencing, primer schemes used. These kinds of things are tremendously helpful for running a good quality analysis, and they are unfortunately still missing from many of the submitted samples nowadays. And that has nothing to do with data privacy concerns or so on. It's just that you need to improve your workflows in-house.
Do I hear a little bit that people maybe sometimes hide a little bit behind the law because there are resource shortages, there's maybe, I don't want to spend the weekends working.
It's a complex topic,
Yeah.
right? And some of these concerns are serious, and I don't want to claim that anyone is hiding behind something, but look into possibilities that you have within your legal framework and see if you can improve things a little bit within these bounds. That's my recommendation. And the other thing, of course, changing those legal bounds, adjusting it to the problem also needs to be addressed, but that's longer term.
Okay, thank you very much. Okay, it's 10 minutes into the coffee break now, so I think I'll let you go. So thank you very much to all the speakers. Thanks for coming, and yeah, enjoy the rest of the day. Bye.